Hello students, today we are continuing with 11.2 and we are looking at muscle contraction. So the essential understanding is right here. It talks about how calcium ions and the proteins tropomyosin and troponin control muscle contraction. Something important for you to realize is that these two proteins, tropomyosin and troponin, are found in the thin filament known as actin. Also, that calcium ions are released via a depolarization of a nerve and through an action potential. So the whole process of muscle contraction occurs over four main key steps. The first one is depolarization and calcium ion release. The second one is actin and myosin cross bridge formation. The third one is the sliding mechanism, or also known as the theory of sliding mechanism of actin and myosin filaments. And the fourth is the sarcomere shortening, which is what involves the muscle contraction. So we're going to start with the first step, which is the depolarization and calcium ion release. And it's a really nice animation here showing you how that happens. So here we see that the action potential was initiated in the motor neuron. And here's the motor neuron. And of course, the motor neuron is connected to your brain, right? To your brain neurons. And this motor neuron, notice how it releases acetylcholine. And acetylcholine is what we know as a neurotransmitter. And it releases it into this cleft known as the synaptic cleft right there. So this is what an action potential is, right? An action potential comes from your brain. It initiates into this motor neuron, and the end of the action potential is what releases the acetylcholine into the synaptic cleft. Once the acetylcholine is released, a depolarization process happens, and here we see it. So the depolarization process is kind of like the impulse spreading, and it spreads along the sarcolemma. Remember that this is the outer covering or membrane, and along the T-tubules right here as you see them highlighted. So then that whole depolarization is going to release calcium ions. And notice that the calcium ions are released from the sarcoplasmic reticulum. And here we see them in green in just a moment. Boom, there it is, right? The calcium ions are being released. And notice that it's that release of calcium ions that is going to trigger the sarcomere, which is the contractile unit, to shorten or contract. So we're going to see it contract in just a moment. Here it goes. Contraction. So notice that when the sarcomere contracts, we see an overlap of the myosin and the actin. Okay? So now we're going to look at step number 2, which is what happens in that overlap of the sarcomere. And here we see that actin and myosin are forming a cross bridge. So let's look a little bit in more detail here at the structure and then the role of this calcium in the cross bridge. So here we see actin. So remember that actin is the thin filament. And here we see a covered actin. And what do we mean by that? Well, we mean that there are these sites on actin that are covered by this other protein molecule called troponin. And because troponin is not activated, if you want to call it like that, then troponin kind of dictates how tropomyosin is going to be aligned along the binding sites of actin. So here we see the two proteins on actin called troponin and tropomyosin, and this is what they look like when the binding sites on actin are covered. Okay? So what happens when calcium ions are released from the sarcoplasmic reticulum? Well, they're going to bind to troponin, and then notice what that does to tropomyosin. It kind of moves the railing, right? It kind of moves the protein, and then it exposes these binding sites on actin. So this is what we called an exposed actin, and it's been exposed because of the calcium ion that just was able to bind to the troponin. Okay? So when that happens, the actin forms a cross bridge with myosin, which is not shown here, but I'm gonna show you in the next picture. Okay. So the next essential understanding is that the contraction of the skeletal muscle is achieved by the sliding of actin and myosin filaments. So this is important. And ATP hydrolysis and cross-bridge formation are necessary for the filaments to be able to slide. So number 3, it's probably the important part here, which is known as the sliding mechanism of actin and myosin. And this is what actually is going to cause the muscle contraction. So I'm going to move it up a little bit. And here you see, it's also known as the sliding filament theory of muscular contraction. And here we see that the calcium ions in green right there have bound to troponin, and tropomyosin has exposed the binding heads. And now myosin, which is here, so this is myosin. Sometimes it's difficult to identify the two. And this is actin, right? And remember from previous videos, I told you that myosin has these binding heads, right? And the binding heads are critical for muscle contraction. So here we see that ATP binds to the myosin heads, and it breaks the cross bridge that was once formed between actin and myosin. So ATP hydrolysis, or the use of ATP, right, turning ATP into ADP, right, causes the myosin heads to move. So here's the energy for the myosin heads to move. And when they move, you see here that they change position and they kind of swivel. And now they're going to go to the next actin binding site here. So here we see that the swiveled myosin that has now headed to the next binding site attaches to the new binding site and then returned to the original configuration. So there's a reorientation that drags the actin along the myosin in a sliding mechanism. So here we see the muscle in motion. It's a nice little animation here that shows how the myosin heads and the actin filaments are moving past each other in a similar fashion as this oar here is propelling a rowboat. And here we see the motion here, right? And it's all hydrolyzed by ATP. Okay? So this is the sliding filament theory of muscular contraction. So once that has happened, that causes the sarcomere to shorten, which is the 4th step. So here we see a 2D representation of a relaxed sarcomere and then a contracted sarcomere. And here we see a 3D representation of a relaxed sarcomere and a contracted sarcomere. So notice that because the actin and the myosin were able to slide past each other, right? And this is because the myosin heads kind of reoriented themselves, right, to kind of like that swiveling motion, right, and that oar rowing that boat. The myosin heads repositioned themselves and they were able to drag the actin filaments past the myosin filaments. Okay? And when that happened, you get this contracted sarcomere, which is what gives you the muscle contraction as a whole. Okay? So here's a nice little summary of the muscle contraction. So remember that it all starts with an action potential, and the motor neuron releases the calcium ions from the sarcoplasmic reticulum. The calcium ion binds to the troponin, which is on actin. It causes the tropomyosin, which is also on actin, to move. It exposes the binding sites on actin so that the myosin heads can now bind to these sites. The actin filament and the myosin heads form a cross bridge, which is then broken by ATP. ATP hydrolysis causes the myosin heads to swivel and change orientation. That swiveled myosin head now binds to the actin filament, and then it kind of swishes that actin filament as it moves to its original conformation. And the reposition of the myosin heads moves the actin filament, of course, and then it contracts the sarcomere. And here is an animation for you to see. Okay, the actin here, the binding sites, notice are covered, but calcium is going to go and bind to troponin, exposed sites, cross bridge, ATP causes the hydrolysis, so that now the myosin heads can kind of move, see, and then they move to the next binding site, and it's kind of like growing. Okay, it's kind of like growing, and that's what allows the muscle contraction. I'll let you watch that in its entirety. Okay. And of course it happens very quickly, very fast process. But it's a repetitive movement. Okay, so that is it for muscle contraction. I hope that you don't have many questions, and if you do, don't hesitate in asking me.